right, amazing. So thanks so much for inviting me. I am Stefano. I'm a professor in the Department of Psychology and Neuroscience. So my main goal is to understand how the human mind and brain work. But I also do it with artificial intelligence tools. And I think that these kinds of tools are really useful to think about how the human mind and brain works. And also, I think that sometimes understanding some things or asking some questions about how humans work can help us gain some inspiration about how to build artificial intelligence systems as well. So my plan for today is to share with you some ideas at the intersection between human intelligence and machine intelligence. And I'd like this to be a conversation. So if you have questions at any point, feel free to kind of jump in and ask. I'd rather, if you have questions, that you ask right away so that we kind of take advantage, like get in the moment, clarify things if they need to be clarified, or address any curiosities you might have. And if we don't get through all of the slides I prepared, it doesn't matter. That's fine, as long as we get to have some nice exchange and you get to ask the things that you're curious about. All right, so I'm going to talk about this from this kind of framework or perspective of NeuroAI, this idea of the intersection between cognitive neuroscience and artificial intelligence. And first, I'm going to tell you a little bit about how artificial intelligence can serve as a tool and as a model to study the brain. These are two quite different uses of AI. One is using AI as a kind of data analysis method or approach that can be used to gain some insights about the mind and brain that otherwise would be hard to obtain. This is more kind of using AI as an applied method for data analysis in the context of neuroscience. But also we can think of AI as a model of what happens in the brain. That's a little bit different from the relationship between AI and some other disciplines, like maybe engineering or chemistry or other things, where in this case, really when we think about humans, right, we are intelligent systems that can get some inputs and then generate some behavior, generate some outputs, right? And so in some way we can then use AI not just as a tool to analyze data, but also to try and model what happens when people are performing all the kinds of tasks we perform. So can we take AI models, get them to do some of the kinds of things that humans do, and then try to start asking questions about if the AI is able to solve some of the problems that humans can solve, are they doing it in some similar ways? Are they using some similar mechanisms? If they differ, what are the differences and why? Okay, then after that, I'm going to try and make the case that in some situations, studying how the brain and mind works can help us to actually develop better AI. And so for that, we're going to talk about some old, early historical examples of this kind of thing. But then afterwards, we're going to talk about some more recent examples of cases in which maybe some ideas based on studying how the brain works can help us to gain inspiration and help us think about AI models. All right. So let's start from using AI as a tool. Let's look at some examples. So I wanted to show you here one example from the study of psychiatric disorders. And one reason to choose this example is that this is a very important problem in society. Psychiatric disorders are very widespread. They affect about 1 in 5 individuals. And they are also extremely, extremely expensive. The estimated cost of psychiatric disorders is $292 billion annually in the United States. Of course, this only captures a small part of what is the world total of these disorders. And just to give you an example how pressing of an issue this is, consider that suicide is the second leading cause of death among individuals in the age bracket 10 to 14. So trying to address psychiatric disorders, develop better interventions is a really, really important, impactful problem. It's also extremely hard. And to explain one of the reasons why it is so hard, I wanted to show you one specific example that is the case of autism spectrum disorder. And if you consider autism spectrum disorder, it has one name. You might feel like it's one thing. But when you start looking a bit more closely, you see that there's many, many different genetic bases for autism. You have more than 40 different copy number variations that are this like genetic indicators that are associated with a higher risk for autism. And different individuals have different CMVs. So it's not like a set of 40 CMVs, and individuals will have those 40, then they tend to be diagnosed with autism, but rather one person may have one, another person may have a different one, another person may have a completely different one. And in many, many of the cases, actually, there isn't any specific copy number variation or genetic basis that is known for a certain individual who is diagnosed with autism. So at the level of genetics, already there's a huge amount of variation, and it's a very complex phenomenon. Now, to make matters worse, this heterogeneity is also observed in neural measures, right? So maybe you might imagine, maybe there's all this genetic variation, it's very complicated, but hopefully then you look at the brain, and then somehow all these different genetic phenomena are all affecting the same underlying pathways, and then maybe they all result in the same kind of neural alterations. That would make things simpler, right? Except that that's not how it works. You still see a huge amount of complexity and heterogeneity at the level of brain anatomy and brain function. And so, for example, you have one particular CMV in autism that is some alteration in the 16p chromosome, and you can have participants with 16p duplication who tend to have smaller brains than average, and individuals who have 16p deletion who have larger brain than average, so completely opposite anatomical effects that are all associated with a higher risk for autism spectrum disorder. Just to give you a sense of how complex the amount of variation that is present. Okay, so we established that psychiatric disorders and, for example, autism are extremely complex. You have a huge amount of individual variation within the same disorder. For this reason, it is very likely that different individuals will need different kinds of support, different kinds of interventions. What may work for one person might not work for a different person if that person has a completely different genetic basis, completely different underlying neuroanatomy and functional alterations. And for this reason, it's really important to try and understand and model this individual variation so that we can improve the diagnosis, try to understand, for one individual who's diagnosed with ASD, what type of ASD are we looking at here? And also to develop personalized interventions so that the intervention can really be targeted and developed specifically for the specific individuals. Okay, so that's kind of what's going on here. That's not good. Oh, all right. Let me see. Oh, this. Hopefully it doesn't ask me to redo this ever. And we are going to be back in a moment. Oh, yes. All right. Let's see. Worst case, we're going to have to do this every 15 minutes, but hopefully not. All right. Okay, so for this reason, we really want to model individual variation and better understand individual differences associated with autism spectrum disorder. And of course, autism spectrum disorder is one example, but a similar kind of problem happens for many, many different kinds of psychiatric disorders. And that's kind of where our AI as a tool is going to come in, right? I sort of took a little bit of a tour from AI for a moment there. Hope I didn't lose you there. And modeling individual variation then is what we're going to be applying AI as a tool for. Okay, great. So this is work that was done largely by my postdoc, Idis, and was published a couple of years ago on Science. Okay, so what do we do? Well, we try to identify individual variation that is associated with autism, right? And one of the reasons why this is a really hard problem, and it took us a little bit to realize this is kind of interesting, where sometimes it takes some time to really understand what is the right question to ask. And then once you figure out what the right question to ask is, then it's sort of faster to sort of figure out a solution. But one of the things that took us a little bit of time to figure out was that one thing that makes this really hard is that not only have a lot of variation among different individuals, but also a lot of this variation isn't actually related to autism at all. Right? Consider a group of individuals who are not diagnosed with autism, and you look at their brains, they're also all different. Right? There's a lot of different dimensions of variation across individuals that aren't specifically related to autism. And so when you go and try to understand individual variation that is relevant to understand autism, you can't just start comparing how different are the brains of different individuals who are diagnosed with autism, because a little bit of those differences would be relevant to autism, but a lot of those differences are going to be differences you'll see also among control participants who are not diagnosed with autism at all, right? And so you have that individual variation is a mixture of variation that is specifically related to ASD and variation that is shared with the general population. And in order to really understand the variation that's relevant to ASD, you need to separate these two different sources of variation. Okay, and so I'm going to illustrate that with an example that's easier to visualize, and then we're going to move to brain anatomy. All right, so this is an example that's easier to visualize with a bunch of pictures, and then some of these pictures are just some like backgrounds. You have whales, like buildings, all kinds of stuff, and then on some of these, you have superimposed some handwritten digits for MNIST. Are you all familiar with MNIST? All right, see a lot of nods. Nice. Okay, and what's the idea here? Well, the idea here is that it's a little bit of an example that has some similarities to our problem of studying individual variation in autism, right? That is, you have that the images that have a blue outline, they kind of correspond to control participants. They all vary among each other. There are all kinds of different one from another. You see that all these background pictures can be all different. But in the case of the green images, the ones with the green outline, you have some additional variation. There is changes in the background, but on top of that, you also have differences in the handwritten digits that are superimposed on top, right? And the idea is that the images outlined in blue, we kind of the equivalent to our control participants that only vary along the dimensions that are shared with all the control group. But the images outlined in green are kind of corresponded to our ASD participants, where you also have variation among those shared dimensions, that's the backgrounds. But in addition, you have some variation along unique dimensions of variations that are, in this case, supposedly ASD-related one, which here in this example are just the differences in the different digits. And the question is, can we build some system that can separate out these different sources of variation and separate out, okay, this is the variation in the backgrounds that is in common across both the images outlined in blue and the ones outlined in green, and then separate out from variation, which is just specific to the images outlined. And so to do that, we use the kind of tool that has been built recently to try and deal with this kind of problems. And it kind of works like this. You take some picture as an input, and then you have some convolutional encoder. So I'm going to say some things, and then if you have questions about things I say, I don't know exactly, what kind of shared knowledge I should assume. So if at any point any of you see some things that are not clear, then just feel free to stop me and ask. Okay, and then this convolutional encoders is going to generate as outputs. So it has these sort of two branches, and later we're going to see why the two branches are important. And each branch is going to generate a vector of means and a vector of standard deviations. Okay, and then what we'll do is for each of these means and standard deviations, we're going to sample some value from a normal distribution with that mean and standard deviation. And then we're going to put it into a vector that we're going to call C. Okay. So we start from the very first mean in this vector of means and very first standard deviation in this vector standard deviation. We sample our first Z, and we put as the first dimension of this vector Zj. And then we do the second one, and so on and so forth. Okay, great. And then we're going to get our whole vector Zj, which is this one. It's like a latent vector. And then we're going to repeat the same thing for our vector Zk using the means and standard deviations here. And then we can use a decoder that is going to reconstruct the original input. Okay, so this essentially is an autoencoder. Because of this business here with the probability sampling is a kind of autoencoder that's called the variational autoencoder. Have you seen variational autoencoders before? Some of you? Okay, a subset, that's fine. It's good. So I also tell you some new things you haven't seen before, because if everyone has seen everything that I'm going to talk about, then that's not such an interesting talk. Okay, great. And so, and as a kind of variational autoencoder also, though it's a little bit special, or it's a little bit different in that normally you just have one branch, right? Whereas here we have these two different branches, right? Now, there's something really important about these branches though, right? Which is we're not going to always use both of the branches
to reconstruct all of the stimuli. Otherwise, there's no point to having two different branches. We may as well just have had only one, right? The important thing is that here what we're going to do is that when our input has both sources of variation, both the one in common with the general population and the specific one, the digits, then we're going to use both of the branches. But if the input only has the background variation, we're going to only use one of the branches, reconstruct, right? So imagine it this way. This blue branch, you can use it to reconstruct all of the pictures, both the ones that have the digit and the ones that don't. But the green branch, you can only use it to reconstruct the pictures that have the digit. And when you train the variation autoencoder in this way, this branch learns to capture information that is more about the background, that is shared across all the inputs, whereas this branch over here, the idea is that it should learn to capture information that is specific to the variation that only occurs among the pictures with the outline green that are used for training that branch. Okay, and then as often happens, you train it to minimize some loss function, which is in this case made of two pieces. One piece is a mean square error loss that tells you how good the reconstruction is. Right? You're comparing the output here to the original input. And the other piece is a piece that is usually what you have in variational autoencoders, which is this term that is called the Kullback-Leibler divergence, which is just a fancy name to say some kind of distance in a way. It's not really a distance because it's a symmetrical distance. They're supposed to be symmetrical, but anyway, some kind of deviation of this distribution that you have here from a prior distribution that has mean zero standard deviation 1 that serves as a regularization term to prevent overfitting. Okay, so then you train the model to optimize this loss. And then you see whether it actually separates the information that is shared across all the pictures from the information that is specific. And you can do this sort of thing. And then you can try to reconstruct images just using the branch for the digits. And you get things where you can get some reconstruction of the digits without the bag. Okay, so the idea is to try to use the strategy to study our problem of individual variation in autism, where we have some individual variation that we expect is going to be there in the ASD participants, but also in controls, but then some individual variation which we expect to be specific to the ASD population. So we're going to have brains from ASD participants, and we have 3D images of their brain anatomy. So here we're going to use a 3D convolutional encoder, and so on. So, and in that case, we're going to try and reconstruct them using both of the branches of the CDA, right? But then we're going to have some brains from control participants, and those we're going to reconstruct them only using one of the branches, right, that we set for the pictures. And after all, we want to check whether this strategy of separating the two branches is actually doing anything useful. And so as a control, we can take just a standard variational autoencoder that has the same structure as this thing. It has the same number of latent dimensions. It's matched the number of parameters and so on, but it doesn't make the differentiation. It's trained with both sets of anatomies. You can use the whole network for all of the inputs, right? And so the idea is that if this strategy of sort of training in this way, in this contrastive way, is really helpful, you should see that this will do a good job at separating information that's specific to autism from variation that is shared with the rest of the population, whereas this one should not be able to do that. Okay, so once we do that, how can we tell if this stuff works, right? Because we can do all of this and then we get our model, and then the question is, okay, that's nice, but does it actually work? And so what we can do is we can try and go and compare individual variation in anatomy based on the latent spaces of these models, and go and compare it to other measures of the participants that we're interested in, right? So for example, for every pair of participants, we can see how similar or how different they are based on their latent representations in the different models, right? So for example, in here, this blue branch is the branch that is capturing variation that is shared among controls and patients. And so we can go and look here, and we expect that the difference between two participants here in anatomy, right, this view here, should capture how different these individuals are along information that's shared across both groups. Whereas in here, this is the one that's only used for the ASD group. So we expect the individual variation in anatomy that's captured here, right? How different two people are based on this to capture something that's more specific about ASD, right? So what we can do is go and take these measures and relate them to other information that we have. For example, we have behavioral measures of symptoms, right? Like ADOS scores, right? That capture the kinds of behavioral symptoms that people are experiencing. And then you can ask questions like, how related is how similar two people are based on behavioral symptoms to how similar they are based on, say, the ASD-specific properties of anatomy. I would expect that if things worked properly, there should be a correlation there, right? Because these are properties of anatomy that should be specifically relevant for ASD. And we're comparing that to similarity in behavioral symptoms that is specific to ASD. Yes, you have a question?
Like specific, I guess, like individuals with ASD, say with eating disorders against like other individuals with ASD to like identify specifically.
Great question. Yes. So this is a really good question because there's a lot of work that has been done to just try to compare the participants overall to controls overall, right? But one of the challenges that we have is really there's so much variation within the ASD, right? And so it's been really, really hard to find something in common to all ASD participants that makes them all different in the same way from control participants. And so what we're doing here is we're looking at individual variation within the ASD group. So rather than saying, can we find something that's in common to all ASD participants that makes them different for control participants, we're thinking, well, there are so many different genetic underlying bases for ASD. There are so many fundamental different changes in anatomy and function. So instead, maybe we may have many different kinds of ASD. And there may not be nothing in common across all of them, except some of the ways in which they manifest behaviorally. And so, yes.
What sort of images are you using here?
Oh, great question. So here what we are using is anatomical images of the brain, and this is usually acquired with an MRI scan or a magnetic resonance imaging scan. It takes usually about 5 minutes. Participants lie down into a scanner, and you can acquire these images at about a 1 millimeter cube of resolution of the brain. They're usually downsampled before going into the autoencoders to some degree. Yeah, that's the type of images that we usually use. And I can tell you more about how MR images are acquired if you'd like. But yeah, you can imagine that's the type of data that we're using. Right. Great. So these 3D MR images of the participants' brains are going in, and we're looking at differences between individuals who are all diagnosed with ASD, but trying to see whether we can capture differences in their brain anatomy that is now related to specifically ASD relevancy. And so our prediction is if this worked, that the individual differences in these anatomical features should be related to differences that are relevant to ASD, more so than the ones here. Now, the individual differences here may be more similar to general variation across people, like variation in age, for example, than the similarity here, right? Because differences in age are also present in the general population. And instead, we expect that here it may be difficult to see a correlation between how similar people are in these features of anatomy and ASD-specific measures of symptoms, because in here we haven't separated out the variation that's specific to ASD from the variation that is shared with controls. And so if those two things are mixed together, we expect that the variation that is shared with control will kind of conceal the variation that is specifically related to ASD and make it hard to see how that specific variation is related to symptoms differences. So the predictions make sense. All right, great. So let's see what happens, right? So here is correlation with similarities based on total ADOS scores. That's a measure of behavioral symptoms of autism. And when you try and compare that to the control VAE model, you can see that the correlation is really around 0. It's really bad. So as expected, right, if you just look at the standard VAE model, you actually don't see a relationship between individual differences in brain anatomy and individual differences in the behavioral symptoms. And we expected that this might be because a lot of the individual variation in anatomy here is actually shared with the general population. It's not the variation that's really specific to autism. Now, when you can look at the similarity based on shared anatomical features or background features and the salient ones, that is the ones that are specific to ASD, you see that the background features don't really correlate either, but the ASD specific ones do. So you see now there is a significant correlation between the individual differences in anatomy that are extracted, the salient ones that are extracted by the CVAE, and the individual differences in symptoms that otherwise you wouldn't see. Now you can do that also with DSM-4, which is another measure of symptoms. You see the diagnostic manual, and you get something similar where you now see that after separating out the ASD-specific anatomical variation, now you have that the individual differences in anatomy that are extracted by the CVAE correlate much more with the individual differences in symptoms. Now we can look at factors that are not specifically related to ASD, like age and gender. And when you look at that, you find that those, after separating, are more correlated with the differences in the background anatomy than in the salient anatomy. So you can see that this seems to be able to separate to some degree individual differences in anatomy that are related to ASD from individual differences that are in common with the general population. Any questions so far? All right, great. So the idea that is that with tools of this sort, then you might be able to go and look at the individual differences among participants diagnosed with ASD and try to use this information to then go and inform possible interventions, right? So for example, if you have to determine, can we predict what kind of intervention is going to be more effective for some individuals rather than for some others, then we're going to want to rely on individual differences that are specifically related to ASD and not be confounded by individual differences that may have nothing to do with ASD at all. All right, so, okay, well, at least it happened at a good moment. If you have questions about then this, in the meantime, I'm going to, just let me know if you have questions so far. Okay, so this is an example of, yes, you have a question.
When you say anatomical differences, what are you talking about? I assume you're not talking about like hippocampus size. Are you talking about?
Great question. Yes. So I have a number of other slides about, getting into the more specific details of this project. I didn't include it because there was a lot of other stuff, but I can tell you a little bit about, other things. So from here, of course, one thing that you might wonder is, okay, what are these anatomical differences actually, right? And one of the challenges that we have sometimes is that this kind of models are a little bit like a black box, right? So you look at the latent space of the model and you got a bunch of numbers, right? But what does this number actually mean? Right? It's really hard to interpret. They are the output of a sequence of complex nonlinear transformations. And, there isn't a very obvious way to just look at numbers and know what it means. But there is one strategy that you can use, which is kind of cool. And so here's a strategy that you can do. Now, what you have is a generative model, right? So you put in an input and you can, generate a reconstruction of the input. But also, you can sample from the latent space. You can sample some latent vectors and generate, brains out of that. Now, one thing that you can do in particular is, actually, let me get back to the visualization of this. Okay, so you can go back to this, right? You have a brain anatomy from an ASD participant, and you use this encoder. You have your latent representation here for the shared features and for the salient features. Okay? Now, what we've done so far is we've reconstructed the original brain of the ASD participant with both sets of features. But what you can also do is turn off the salient features and reconstruct the brain using only the shared features. right? Let's think about what that means for a second. It's kind of like we're generating a simulated twin brain of that participant that does not have autism, right? It's a brain that matches the brain of that participant along all of the shared features, but doesn't have any of the ASD specific features, right? Now, this is really useful because what you can do is you can compare these two brains, right? And these two brains are matched on the shared features, and so their only difference is driven by the ASD specific features. And so you can take, the original participant's brain, and you can take the simulated control twin and generate a nonlinear morphing that morphs one brain into the other. And so when you do that, you can basically get a vector field that basically for each location in 3D space tells you how you have to move that location to morph from the control brain to the ASD brain. Right? That tells you basically how the brain changes as you go from the control brain to the ASD brain. And so you have one of these vectors for each location in space, and that gives you a vector field across the whole brain. And then from there, you can start doing some analysis on what these vector fields look like. For example, you can take the determinant of the Jacobian of the vector field, and that tells you locally have some expansion or contraction. If the determinant of the Jacobian of the vector field is greater than 1, then it's telling you there's an expansion there. Instead, if it's smaller than 1, then it tells you there is a contraction, right? And so you can generate whole brain maps of expansion and contraction across the brain as you go from the control brain to the ASD brain. And that enables you to start identifying which brain regions are affected and how. And so, sorry, it was a bit of a long-winded answer to your question, right? But that's how you get to, going from, this is a bunch of numbers that we don't know what they mean in here, right? That came out of this nonlinear thing that we don't know how it works, to, hey, in this particular participant, ASD-specific features generated a contraction of the set of brain areas and expansion of this set of brain areas. Now you can interpret what they mean going back to other neuroscience findings in the literature, what they discuss about what is the function of those areas and how it's relevant to behavior. And so that's how you can make it interpretable and relate to some of the rest of the work. Yes?
To get from the MR images to the flow to the vector field you're mentioning, you would have to compute the flow rate. Because the
Yes.
MR images are just like.
Yes. So you have two images, right? So you're going to have, you're absolutely right. So the procedure works like this. So you have one image, which is your reconstruction from the autoencoder of the original brain using all of the images. Now you have a second image, which is the reconstruction that turns off the ASD specific features. It uses only this. Right? So then you have two images. One is the reconstruction of the original input, and one is the reconstruction using only the control features. And now your vector field is computed as the vector field that you need to transform one into the other. So we have algorithms for morphing brains from one brain to another. They've been around for quite a while because these originally were developed because people wanted to do some group analysis. They wanted to get data in many different participants and then try to do some statistics across, say, 20 different people to see, is there brain activity in this place across all these 20 people? But the problem was that when they were trying to ask these kinds of questions, they run into the issue that every different person's brain has a different shape. And so they know how to compare them. So they decided to do in this way, to come up with some template brains. And so they made this, template brains. One is known as the Talairac brain. Another one is the MNI. That stands for Montreal Neurological Institute. So they're these template brains. And what people would do is they would take each individual person's brain that has a different shape and do a nonlinear transformation that morphs that into the template brain. So they take all these different brains that are all different shapes, and they would morph them all into the template brain, and then average results across participants in that way, or compare across multiple participants in that way. So we already had around algorithms to do this thing, morph one brain of one shape into another. In this case, we're using it to morph the control brain into the ASD brain, where they're matched in the shared features, but they differ in the ASD features. And this algorithm, that is the algorithm that gives you the flow, right? So it tells you, here's how you have to move around the points, to morph one brain into the other point, and it gives you the vector. So that was kind of, in a way, easier. We could kind of reuse some of the hard work that other people had done before us. Just take it, get it to give us the flows, and then be able to calculate our statistics on that. So that's how you can go from, having it as a kind of black box, things where you have all these latent numbers that are hard to interpret, onto knowing, what kind of changes are happening to what brain regions. Great questions. Other questions so far before I go to the next part? No? Think about it while I reconnect to the system. I wonder why the system has so little trust in me, but all right. So, okay, great. Okay, so let's talk about AI as a model. All right. That's a little bit of a different way of using AI to try and think about the mind and brain. So, okay, suppose that you have a picture, and then the picture, you can show it to a person, it goes into a brain, and then the brain says, oh, that's Stefano. I know that. Okay, so we can recognize people, retrieve their names. All right. Well, ideally, say they want to try and understand how this kind of process works. What is happening in the brain as, you see a picture, and then, some stuff happens, and then you recognize who that is, right? And the idea is you can take some similar process where you take a picture and you show it to, say, an artificial neural network that has been trained to do something similar to recognize, okay, who the person is based on the picture. And in the end, you can ask questions such as, is the stuff that is happening in here in the neural network in some way similar or different from what is happening in here in the brain? If it's similar, in what ways is it similar? If it's different, in what ways is it different, and why, right? And it is that hopefully going through this process can help us gain some deeper understanding about what is going on in here when we recognize a picture. For example, now this is just an example of it, right? It's not just recognizing a picture. You could have it for like recognizing the genre of music, or understanding a sentence. You could use the same kind of strategy for all kinds of things there. Okay, great. So now one of the challenges we have to deal with when we do something of this sort is figure out how we compare what's in here to what's in there, right? Because they're very, very different things, right? In there, you can have many different layers of a neural network. You can get the activations of the units in the different layers. And here you can measure, sometimes the activity of individual neurons. Sometimes you may measure the aggregate activity in larger groups of neurons, like what you do with functional MRI. But there isn't a very trivial way to compare one-to-one, right? It's not like, one hidden unit in the network fully corresponds to a neuron in the brain. And so we have to deal in some way with the issue of how do we even compare these two things? It's kind of like comparing apples and oranges. We need to kind of find some kind of common language to compare these two things. And so the idea is to try to capture what kind of information is encoded in a brain region and in a layer of a neural network in a way that makes it more comparable. So let's see one way that we can try to do something of this sort. So if we take some brain region that we think is important for recognizing pictures, this is ventral temporal cortex. We know that animals and people have lesions there. They have deficits for image recognition. So we know that's important. Say that we take this brain region, we measure the responses there, and then we show someone a picture, say the picture of that face. And what we're going to do is we're going to be measuring the amount of response in many different locations in this brain region. And we can visualize that as a vector where each dimension corresponds to a different location in this region and has a certain amount of response to that image. Now, what we can do is now we show a different picture, and we get the response in that region now to that picture. And now what we can do is we can make a little table here, a little matrix, where we're going to encode the similarity of the responses to the different pictures. So I'm not filling here the diagonal because it's kind of trivial. This, is the same response. So, the response to the same picture. So it's going to be obviously very similar. But here we can encode basically how similar is the response by 1 to 1 picture and the other. And there's various ways of calculating the similarity. People can use Euclidean distance or correlation distance, which is not really distance. But anyway, Malanobis. There's a whole variety of metrics that you can use to do this sort of stuff. And if you're interested, then we can talk more about that and why one might want to use some metrics or others. But for now, I'll kind of ignore the issue and keep going with the main focus, which is capturing the information that's encoded in this region. Right now, we can show say another picture. Now we should say of a house. Now we get a pattern of response. Say that's now quite different pattern of response, and then we can encode it again in our table of how similar it is to the responses to the other stimuli. All right, and so in here we basically got a table or a matrix that tells us for each pair of stimuli how similar are the brain responses in that region to those different pairs of stimuli. Now, this is quite nice because, so this strategy is called representational similarity analysis, or RSA, and it's quite nice because now we can do something similar for layers from neural networks. And although, the amount of neurons in brain region and the amount of units in the neural network are very different, they're not even one-to-one comparable in any meaningful way. In the end, we end up getting matrices that are just number of stimuli by number of stimuli. And so those are more easily comparable. So we can take your neural network, look at a layer, and do the same thing. You get the amount of response in different units to the stimulus, to the other stimulus. You make your table. And now what you end up having is you're going to have one of these things for your layer from the neural network, and one of those for your brain regions. And as long as you show the same set of stimuli to your neural network as you did to your participant, now you can compare these two and see how similar or different they are. If it turns out that your model is doing something that is in some ways similar to what is happening in the brain, then you would expect that this would be potentially more similar to what you see here. And instead, if they have very, very different things going on, then you may expect that they're not going to be as similar. Of course, them being similar doesn't guarantee that the brain and the body is doing exactly the same thing, right? It's just telling you it's more likely that this is a good candidate model versus a bad candidate model if it's kind of more similar versus more different. Okay, great. Any questions so far? Okay, great. All right, so people have been using this kind of strategies to compare artificial neural networks to brain activity in a number of domains. I'm just going to show you one example here, and then we're going to see another one later on in more detail. But this is in the case of visual object recognition, for example, but people have done something similar for auditory perception and many other things. And this is just when people were trying to model the responses in a brain region that is considered a sort of high-level vision region. And it was really hard to model with simple features that people tried with all kinds of, hand-designed features. But it turned out that actually they could do a pretty good job using artificial neural network features. And what you see is that using just the kind of strategy that I told you earlier, by looking at the correspondence between brain activity in this higher level later regions in the visual system and their responses in the artificial neural networks trying to recognize objects, you see that these later layers of the neural network show the particularly high correlation with the neural responses, whereas the early layers didn't do very well. And this gray band that you see here is a thing called the noise ceiling. Basically, we want to get a sense of how well we can possibly do, and the way this is calculated is usually by comparing data from different people or from different animals. So, for example, say you have, I don't know, data from 10 monkeys, right? Say you take the average of the responses of the similarity matrix that we showed you earlier from 9 monkeys, and we see how similar that is to the remaining 10 monkeys. Now, we are never going to be able to do better than that with the model, right? Because the model isn't built to capture individual variation, right? You're going to use always the same model-based similarity matrix, right? So it's not individual specific. And so all of the variation that isn't even consistent among humans or among monkeys, you can't capture that with the model built in this way. You have to build a specialized model for each individual, right? And we didn't do that, right? So seeing how much correspondence you have between people or between monkeys gives you a noise ceiling that tells you that's how well you can possibly do with your models, right? And then you can use it as a sense to see how well you're doing. And you can see that with these later layers, you can actually do pretty well. It's quite interesting, actually. If you look at very early visual regions, you get the opposite pattern where regions that are very early in the visual system actually are better captured by the early layers in the neural network than by the later layers. So you see some degree of similarity between the hierarchy of processing in the human visual system and the hierarchy of processing inside the layers of the convolutional layer. Okay, any questions so far? Right, awesome. So this is an example of using neural networks as a model of the visual system, right? Because here, this is not just a tool to analyze data, but really we are thinking that, we're asking people and networks to do similar kind of tasks with similar kind of stimuli and trying to compare what happens inside the brain and what is happening in the networks. All right, how does this help us to understand the brain, right? That's an important question, right? That many people have asked in the field. Okay, so now we have this model. So what? So how does this help us to understand the brain, right? Just predicting something doesn't necessarily mean that we understand it, right? And so here the important idea is that what you can do is start comparing many different sorts of models and seeing how well different kinds of models match brain activity. And once you start seeing some differences, you can start asking, why is it that some models are better at capturing what happens in the brain and some models are worse? What is driving this difference in performance? And that is what can start to help you really make some progress in understanding what may be going on in the brain and what kinds of computations it might be. Any questions so far? All right. So, okay, so this was more about how we can use AI to study the brain and mind, right? As a tool and as a model. Now, I want to make the case that studying the brain can help us develop better AI. I also realized that we don't have a lot of time because we had a lot of very nice conversations about the first part. So I might do a bit of a selection and tell you some of the things that I was planning to say, and then you can ask me questions along the way. So for early examples, okay, so there's some old classical examples of how inspiration from the brain has helped to develop AI. Probably the most classical ones I just touched upon it briefly is this experiment by Hubel and Wiesel. Have you heard of this stuff before? I don't know, it's like a really neuroscience people stuff. So cat under anesthesia. Yes, exactly, a cat under anesthesia. And so basically Hubel and Wiesel were trying to figure out how the human visual system works or how the animal visual system works, right? And so they were looking at single cell recordings from early visual cortex, an area called D1, and trying to figure out what drives the responses in these neurons in the D1, in this case of cats, right? And they tried to show all kinds of things, and it seemed to be really hard to figure out what was driving the responses in these neurons until at some point they noticed that there was a strange phenomenon where they had the cat there and they had the recording, and they were using this really old projector. Like, I don't know if you've ever seen stuff like that. It's like, it wasn't even almost around anymore when I was a kid. So it's really ancient, right? So it was this projector that's using like this kind of transparent sheets, and it has a light that shines underneath. And you can put like these sheets with some drawings on it, and you can slide them on, and then it projects the drawing on some screen or some. All right. So they were using that kind of projector, and they noticed that at some point they had this case where the things that were showing to the cat, they really weren't driving the responses all that much. But whenever they put in a new sheet or took out one of the sheets that was like there on the projector, then they had responses. And so they were like, well, what's going on, right? And then they noticed, well, that's because when you do that, you create like a line between the part where the sheet is still on and the part where it's not, right? And so they started trying to use oriented lines and said, well, maybe, that's what's driving responses in this neurons. And in fact, after a bit of trying, they realized indeed that was the case. They could find that these neurons were responding to oriented lines, but in particular for each specific neuron, the lines had to be at a very particular orientation. That was the preferred orientation. It would have like be selected for a certain orientation. but also the line had to appear in a certain portion of the visual field. So there could be, say, a neuron that is specifically selective to, say, lines at this angle that are happening in the top right corner. And if you keep the same angle, but you're showing the bottom left corner, the neuron doesn't respond. Okay. And so they found that there was kind of like a timing where you'd imagine that for each location in the image, you'd have neurons that were selective for all kinds of orientations. And then for each orientation, you'd have many different neurons that are selective for that orientation and many different locations in your visual. Okay. And so in a way, if you want to kind of plot it on a brain, what this looks like. So this is primary visual cortex P1. Here's eccentricity from the center of the visual field out to the periphery. And if you plot it on the brain, you have that neurons that are over here are responding more to things that are the center of the visual field. And as you go towards this direction, they respond more and more to things that are out in the periphery. Now, why does it matter for AI? Well, this is the idea that then gave rise to the idea of convolutional networks. Are you familiar with convolutional neural networks? Well, it's this thing, right, that inspired people to build convolutional neural networks, because if you can think of a neuron that extracts, that is selective for, say, a certain line with a certain orientation, you can think of it as a kernel in your convolutional network. And you have that it's local, like a kernel of a convolutional neural net, that, if you have a convolutional neural net, when you apply the kernel to a certain location of your image, it doesn't care about what's happening in other parts of the image, right? And then you repeat that, you tile your whole input with that, right? So you have that, for example, for the vertical lines, you're going to have a little local vertical line extractor in many different locations. That would be like applying a certain kernel and sliding over all your inputs, and then so on and so forth for all the other orientation, right? So for the vertical orientation, it would be like having it being like one kernel that gives you one output channel. Then you have, say a slightly different orientation. It's now a different kernel that gives you another output channel. And so really this kind of thing, right, it's kind of very similar to the sort of stuff that we see. All right. So I'm gonna skip to stream networks for video processing because we don't have as much time. And for recent examples, maybe I'll just show you 2 out of the 3. And one, very quickly, this idea of adversarial attacks. Have you seen adversarial attacks before? A couple of you have seen this. Okay, so the idea is that you can take a neural network and get it to be really good at some task. I always use object recognition just because it's such an intuitive and easy task to think about. But you could do the same for other kinds of tasks. So you could take, say, some picture like this, which is a panda, and you give it to a neural network that's trying to recognize objects, and it guesses that it's a panda. I said, okay, I have a 57.7% confidence that this is a panda. Right? Now, what you can do is you can train another network to generate some little bit of noise that you can add to the picture to trick the first network. Okay. So for example, in this case, you can add just a little tiny times this kind of noise patterns that corresponds to what you might expect for something that's classified as a nematode. And you get this picture, which kind of looks like the first picture, right? If you look at these 2 pictures, they're not all that different. And it makes sense, right? Because you add a tiny amount, a tiny number that multiplies this. Right? So you change your original pictures very, very slightly. And for us, it kind of still looks like a panda, right? But if you now show it to the network says this is a gibbon with 99.3% confidence, which clearly it's not, right? Now, so what's the cognitive science, or what's the neuroscience part about this? Well, it's not so surprising in itself, the fact that you can break the network by changing what the image looks like. Right? That's not the surprising part about this fact, right? The surprising part is that you can do it in a way where, to us humans, this basically looks like the first thing, right? If you added something and you told me, well, now you get it wrong, and this is completely different from the original one, you'd be like, yeah, well, big deal, of course. If you turn the thing into a gibbon, then the network's gonna guess gibbon, right? But the amazing thing here is that you can do this by turning this into a thing that, to us, it just kind of looks like the first thing, right? To us, like, how's even different, right? So that's where the human, aspect comes into this, is that the surprising aspect of all this is that to us, they still look the same. And so that is telling us that the way our visual system works is still quite different from the way that these kinds of networks work. Because if it worked the same, we should be looking at this and saying, this is a given with 99.3% confidence, right? And we aren't clearly saying that. And so that tells us here there's a big difference between the way these neural networks work and how humans are perceiving this. And so that has inspired a whole field of research into robustness of neural network to adversarial attacks. How can we make models that do not fall into this kind of tricks? And in what way is the human visual system able to pay this sort of mistakes? Okay, I'm gonna skip this one because we're short on time. All right, and I'm going to show you this last one thing, which is a very, very recent example of how you can use some of these ideas from the brain and maybe use them to give some ideas about AI. And this is from some very recent work that was done by a graduate student in the lab, Ahmed, and I have pictures of him in a couple slides. And that is just was just accepted in Cambridge. And so here we have this phenomenon in the literature that's comparing brain activity to deep network models, where people have reported that, in particular, when you look at vision models, models that are better and better at the tasks they're trained with, for example, say at object categorization, they're also better and better at explaining variance in the brain. So across many, many models, people tried, models that tended to perform better, they also seem to capture more variance in the brain. And so one of the things that we asked was, well, recently there are all these vision transformer models that are coming out that are really powerful. Let's see how they compare to the brain. Are they similar or different? And are you familiar with transformer architectures? A little bit. I see some nods. Okay, well, so transformer architectures, I'm not going to get into the details because it takes a bit long to get into all the specifics. But the general idea is that they're really good at capturing relations between things. And so they're often used for language, or the things inside ChatGPT and LLMs and those kinds of things, because you can use them to capture relations between different words in a sentence. But you can do this sort of things also for visual stimuli, where you can take an image, divide it up into quadrants, and then start to build, treat each quadrant of the image like a word, and then process it with a transformer. Okay, I'm not going to get into the details of how the architecture works because we're a bit short in time. Now, recently there's a whole family of transformers, of vision transformers, that have been performing really, really well across many, many different vision tasks that are trained with this trick that is called mask autoencoding, where you take some input and you kind of remove some parts of it. And then you train the model to reconstruct the missing pieces. Right? That's why it's called a masked autoencoder. It's kind of like an autoencoder, but the input is masked. And it turns out that when you train models to do this kind of thing on a very, very big data set of pictures, then they're amazing at doing a lot of other vision tasks. Like, for example, now you can take the latent states of these transformers and try to use them to recognize the objects. And they do extremely well at that, right? They do better than many of the models that were just trained from the get-go to recognize what kind of objects you have in pictures. You can do the same sort of thing also for videos. So you take videos and then you mask some parts of the videos across space and time, and you train to reconstruct the whole video. Often for this, one strategy that's very effective. This strategy is called tube masking, where instead of just randomly masking different parts of the video at each different frame, you sort of like pick a region of the frame that you want to mask, and you mask that part of space across many different times. That's why it's called tube masking. It's like a pipe or a tube that you're masking away. Okay, and so you can do this, and again, these kinds of models are really good at a lot of video processing tasks. So an obvious question is, now that these models get so good at all these different vision tasks, how do they compare to the brain? Are they now even more similar to the brain? And so Ahmed in the lab has been asking this question. And the fun part is, I'm going to just summarize it for you because you imagine we're running out of time, is that they actually don't. They're actually not that great. And they kind of break the rule that better models are always also better at predicting the brain. But of course, the interesting question, so see, here we get a bit to what we were saying earlier, right? When we talked about comparing models to the brain, what is interesting is trying to understand how different models compare to the brain and why are some better than others, right? And so we started asking this question and saying, first of all, let's see whether there are some models that explain unique variance in neural responses that other models cannot capture. And so we calculated these kind of matrices for different separate brain regions, but the pattern is very similar across all these different regions that we looked at. And here what we did is that, for each column here, you have a model. And for each row, you have a model that we controlled for. That is, if you see, for example, this bright yellow little square here, it means that this model here for this column explained additional variation in linear responses after controlling for this model here in the row. So it starts to tell us which models explain unique additional variation in the brain that is not accounted for by other models. And you start seeing some patterns. You have this little square over here across all the regions. And this is telling you that these models, the video models, video muscle encoders, explain unique variation that is not captured by the image-based masked out encoders. So this is a similar architecture. They're both masked out encoders, but adding the video information enables you to capture unique neural variance in responses that isn't captured by the image ones. You also see this little pattern here, and that is telling you that this model here, which was just a convolutional neural net trained to do object classification with ImageNet, explains unique variation that is not captured by the image-based masked out encoders, suggesting that these vision transformers, despite being so powerful, they seem to have something different in their architecture that makes them miss some of the neural variation. And also the big one, of course, right? You may have been wondering about that giant stripe in the middle. Why does he not talk about it, right? That right corresponds to models that were trained based on video information to compute optic flow. And these seem to account for unique variation in brain responses above and beyond all the other models except from themselves. The reason why here it's not yellow is this is themselves, right? This is the same two. And so of course, when you control for themselves, then there's not much left. But compared to all the other ones. And so we started asking, why is it that these models are capturing this unique neural variation that isn't really captured well by the other models? So what we tried to do is we took all the different layers of all these different models, and for each layer, you can calculate one of those big representational similarity matrices that I showed you before. And then what you can do is you can calculate principal component analysis to identify dimensions that account for most variation among the representational similarity matrices of different layers in different models. Kind of get a map of different kinds of representational structures you have in different layers. And so that's what you see here is the first principal, two big principal components. And it kind of looks like you have this bunch of models that are all clustered here. And then you have a few models that are kind of shooting way and far away in this direction, right? This green one, this red one, and this purple one. And those actually turn out to be specifically the only three ones that are the optic flow trained models. So you can see that they have layers that are way out here in this region of this PCA space that is very different from all the other models. And those are the ones that also capture unique neural variation, right? And so, because these layers all had this kind of larger value across the second principal component, we started asking, what does that correspond to? Can we make it interpretable? Can we go and see, when two different frames of our video have a high loading on this principal component, what makes them different? Right? Can we find some interpretable relationship? And what we found was that this is like showing pairs of frames, one frame, another frame, that had very different values along that component. And those tend to be cases where there's a big difference in the amount of optic flow between one frame and the other, really suggesting that there's something different about how these models represent optic flow information compared to all the other models. The vision transformers, even the ones that use video information, don't spontaneously learn how to do this. And these optic flow representations make these models able to capture unique variation in neural responses that isn't quite captured by the vision transformers. All right, so what does this mean, right? So we found that vision transformers are maybe not as good as we expected at capturing brain responses. And we found that there was something about how some optic flow-based models process information that made them more similar to neural representations. Okay, but why? Why is this happening? Now, we don't know yet, right? We need to do more tests. This is just very fresh. But our suspicion is that there's going to be some tasks out there that humans are really good at, that the vision transformers, even the ones using videos, are not that good at. They might just not be among the pool of tasks that people are using right now as vision transformers, but it could be things like, say that someone throws a baseball bat at you and you have to catch it, right, or something like that. Something that involves information about optic flow, rapid dynamics of objects. They may not be captured by tasks that ask questions like, what kind of object is this one, right? Or what kind of action, what category of action is this one, but more like, what trajectory is this going towards, right? So that I can, grab it right away, right? And a lot of these kinds of tasks, they're not being tested for this vision transformer model. So our prediction is that those kinds of models, we expect them to be not so good at some of those tasks, and we expect humans to be really good at it, and we expect that those optic flow representations of that information is going to be important to perform well at those other tasks. All right, so this is just an idea of how sometimes comparing models to the brain, it's useful to try and understand what's happening in the brain, but also it could give us some ideas or suggestions about are we missing something in the AI literature? Are there maybe some other tasks that we should be testing? Or maybe there may be some different changes in the architecture that we should be trying out. Okay. All right, I'm just gonna skip this because I think we talked about this already. Broader take-home points for today. The first one is I told you about how AI can serve as a tool and as a model for doing cognitive neuroscience research. But AI does not have to be like the brain or brain-like to be effective. It's not a requirement, right? You can potentially build AIs that are completely different from the brain and maybe they're still good. But sometimes understanding the human brain or thinking about how it works can offer us some idea on how to improve AI. All right, that was it. Thanks for listening. I hope you had fun. And yeah, if you have any questions, you can feel free to reach out. All right, maybe later. Okay. The thing also stopped disconnecting it. That's nice.
Okay. Or Michaela, you have recording.